Hello, everyone. Welcome to Reason with Science. I'm your host, Jitendra. This is a conversation with Aaron Engelhart. He is an assistant professor in the University of Minnesota, Department of Genetics, Cell Biology, and Development. His research mainly focuses on understanding nucleic acid folding and function. In this conversation, we talk about life on Mars, astrobiology, RNA world hypothesis, and tools to use to find life on Mars. Enjoy the conversation, share, and subscribe to support the podcast. Thank you for listening. Hi, Aaron. Welcome to the podcast.
Hi, nice to be here.
Yeah, so let's start with this crucial question. What are the major or what are the leading theories in origins of life on Earth?
So it's an interesting question because I think that as you might have experienced, everyone will probably give you a different answer. The way that we approach it, I guess I would think about the chemical approach to it, or sort of how we got to where we are now in that respect. Then I guess also the geochemical environment. And so those are kind of related questions, which it was, but distinct in their own way. And I think that on the chemical side, there is definitely a sort of camp that's been sort of RNA first for a long time. And I think the sense I've gotten, having worked in this field, for the past 15 or 20 years, is that there definitely was a sort of contingent that was really RNA first, and I guess you would call it kind of a hard RNA world. And I get the sense over the last 10 years, maybe a little bit more, that there's been a little bit of a softening of that to thinking about, there probably was a stage in life where RNA catalysis was important and more important than it is right now. And I think that we see relics of that in contemporary metabolism. There are ribozymes like hammerhead ribozymes. There's a ribozyme catalyst at the core of the ribosome. The group 1 intron, is one example. But obviously proteins have kind of supplanted RNA as the main catalytic biopolymer of life. And so, in terms of people debating whether RNA or protein came first, I think that the answer is both, with RNA probably being a little bit more important than it was certainly in the past, at present. So I think that it's a really interesting question, and one that can inform our understanding of biology today, to think about ways that RNA and protein can interact synergistically. And coming back to the example of modern biology, I think we see quite a few now. If you look at the ribosome, if you look at the core of it, it's essentially a ribozyme, but there's proteins that are critical for its function as well. And there's countless examples in biology of RNA protein interactions or riboprotein complexes that are really important. And then I guess the second part I mentioned, in terms of a geochemical environment in which life got started, and that's one that I'm sort of agnostic on. I think that people come back to this idea that Darwin had of a warm little pond where there's kind of dehydrating pools where molecules got concentrated. And I think there's a lot to like about that, because you can imagine things like wet dry cycles and energy being introduced intermittently. By intermittently, I mean day-night cycles and seasons and things like that. But then I think there's other environments that are also interesting as well. Looking at things like hydrothermal vents. There's a lot of interesting things happening there. If you look at constrained environments with thermal gradients, there's things like thermophoresis that can be happening. You've got flow there too that can induce mixing. You've got a lot of energy going into it. I think at times it looks like it might be too much energy in some ways, because if you're in a really hot environment, fragile molecules like RNA are going to get degraded to a significant extent. And so I think that, in thinking about the geochemical environment in which life started, that framing, I don't know, is necessarily the most productive one, because if you look at these other environments, there's, like I said, things to like about each of them. And in considering the alternatives, I think that the way that we approach it is thinking about, what can get done in X environment versus Y environment. And you've got, a whole planet or multiple planets to look at in terms of an environment where life got started. And because of that, I think that there could be niches for doing synthesis of some of these protobiomolecules.
Yeah. And what are the major questions that your group is working on?
We are interested in how life could get started, and in particular, looking at ways in which those types of synergistic interactions between different biomolecules can impart interesting functions. If you look at life today, if you look at how biochemistry is studied most of the time, we kind of approach it in a reductive way. And I don't really say that critically. I think that it's been a really productive approach to kind of trying to solve problems of biochemistry. We've learned a lot and continue to learn a lot working with purified biomolecules. But of course, the inside of a cell isn't purified biomolecules. Biology is scaffolded. If you look at the cell, there's subcellular localization of lots of different compartments. And this goes even beyond, kind of what we traditionally think of as organelles. I mentioned the example of RNA and proteins coming together in condensates and things like that. And, we're increasingly appreciating their importance in different biological systems. And I think that it's a really interesting question of how that feeds into life getting started, because what we've seen is, things like operating different catalysts in the presence of different membranes can allow for different biochemical transformations to take place. RNA and proteins interacting. We've done some work. There's been some really nice work from Chris Keating's group on this as well. And kind of looking at those sorts of synergies and interplay between biomolecules is something we're really interested in. And then tying into kind of the second aspect of the framing of the question, how did life get started? The geochemical environment, that's something we're really interested in as well, because I think that that would be really key to what is sort of allowed in terms of chemistry that you can do as life is getting started. And so if you look at, for example, a warm little pond that's drying out or a hydrothermal vent, you've got different temperature conditions and different conditions of things like salinity, and that's all going to influence, what chemistry is possible, but also the preferred folding state of things like an RNA, whether it's folded up or what it is folded up into, and kind of the same for the protein. And I think that, studies of that inform both the emergence of life on Earth as well as, other environments. If you look at places like the modern surface of Mars, it's a really interesting question whether life existed or currently exists. I think an equally interesting question is, what if both of those turn out to be false? On one hand, I think that would be kind of a bummer in the respect that there's, not life there and that looked like a good candidate, but it also is a great candidate for the same reasons that we think it looks like a good place to host life for prebiotic chemistry to happen. And looking at, the surface of modern Mars and the regolith, it looks like there's, quite a few salts there and some really briny environments. And that has its own influence on how different biomolecules fold, what sort of chemistry is possible. And I think that's another case in which we're kind of interested in working at interfaces of, biomolecules with each other as well as biomolecules with their environment.
Yeah, and since you already mentioned Mars, so how's your work is associated with NASA?
So I have been involved with research that's funded through NASA's exobiology and astrobiology programs, since my training labs. I trained in a couple labs that were funded through the exobiology program and their Center for the Origin of Life. And then currently my lab is funded through NASA through a center called the Center for the Origin of Life, that's tasked with studying the emergence of the earliest biomolecules and the emergence of the ribosome and its evolution. And then, I guess that's our principal interaction with NASA in terms of funding. And then beyond that, NASA is intimately involved with the astrobiology community in supporting and attending sort of a number of our key meetings. There is a group called the International Society for the Study of the Origin of Life. And we just had our meeting recently this past year that NASA is involved in supporting. Coming up in May, there's a meeting called the Astrobiology Science Conference, or AbSciCon, that's taking place in Atlanta in May 2022. And they're very involved with that. And then beyond that, they've started a program in the last few years called RCNs, or Research Coordination Networks. And the idea with these are to kind of encourage cross-pollination between different teams that are working on these projects. I mentioned interfaces, and we're talking about scientific interfaces, but interfaces between groups are really important, and this is why we come together at a lot of these meetings. The RCNs are intended to encourage collaboration and sort of this cross-pollination between other groups that are studying it. And there's a lot of groups doing, I think, some really exciting work in this space. And so NASA is kind of instrumental in sort of orchestrating some of these interactions.
Yeah, with that, can you please also mention what are the possibilities of life, coming to Earth from other planets, like Mars, for example?
Yeah, I think it's a really interesting one. And it's, if we look at different examples of how that might have happened, a lot of it was long enough ago that it's kind of difficult to do the experiment or study exactly how it might have happened. But the example that a lot of people go to that's a really powerful one is there is a meteorite that landed in Australia called the Murchison meteorite that landed a few decades ago there. And, or I say landed, it was really crashed into Earth a few decades ago there. And it did, what the Murchison meteorite is a class of meteorite called the carbonaceous chondrite. And it contains the carbonaceous, as the carbonaceous implies, a number of different components that are found in contemporary biochemistry. And the fact that this has gone through that type of transit and that these biomolecules survived, I think is a really powerful example of showing that it's possible for these things to kind of undergo that type of transit.
Yeah, so interestingly, as you mentioned, that there were biomolecules that they came with the meteorite, right? So one thing is that we get building blocks. The other thing is that we get some sort of primitive life forms already. Is it possible that some sort of these life forms, they can survive that sort of conditions when meteorite is hitting Earth, for example?
Yeah, and it's certainly a heavy lift in terms of radiation and the temperature excursions that something like that experiences along the course of that. And it's a little bit outside of what it's pretty far afield from what we study, but there definitely are groups that consider this as a very real possibility. I think that from our perspective and thinking about what's prebiotic chemistry versus modern biochemistry. And if we assume that something did were to come, if it came today on the carbonaceous meteorite, and it was alive, it would be pretty difficult to answer, whether that actually was extraterrestrial life versus life like we find on Earth, precisely because we have a lot of life on Earth, and this is something that we think about in terms of exactly how we study, putting a lander on Mars if life is present there. How do we know that it wasn't what you call forward contamination or brought from Earth? There are certainly ways to do this. If we look at the elements that make up the biomolecules that we have on Earth versus other planets, there's different sort of signature associated with these elements in that they have different what's called isotopic composition. So there's, just like there are radioisotopes that we use to study biochemistry, there are also a lot of stable isotopes that make up sort of modern chemistry on Earth. So like, for example, most of carbon on Earth is carbon-12, but there's a significant fraction, as you know, of C13 that's about 1.1% of the carbon that's present. And this can vary from body to body. And so you can use that to probe whether something's of extraterrestrial origin. But in terms of kind of our core molecular biology experiments, in terms of doing something like amplifying DNA or sequencing DNA or things like that, it's, assuming it is the same biochemistry from something that came from an extraterrestrial body, it's very difficult to sort of discern whether something was of extraterrestrial origin or of Earth-based origins. So it's an interesting question and definitely one worth asking, but certainly one that is challenging and keeps a lot of people going to work and studying this.
Yeah, but then maybe we can zoom in a little bit in this similarities of geochemistry between Mars and Earth, for example, because that's where we can already start saying that, okay, maybe the environment is somewhat similar. So probably at the end, it won't even matter whether the life, either it goes from Earth to Mars or or if it comes from Mars to Earth, right?
I think you're right, and it's certainly possible to have the situation where you kind of do the same experiment twice and end up with the same outcome, because as you point out, if you're looking at the geochemistry of Mars and Earth, there's a lot of similarities. A lot of the isotopes, the same elements are present on both bodies, and it certainly makes it possible to end up with the same solution. I don't think that's necessarily a guarantee. Life has ended up with a lot of things, but they're kind of sort of locked in. The ribosome is a great example. Moving past the very early stuff like the biochemical components, but just things that are similar between all of life. If you look at the core of the ribosome, so the machine in cells that makes protein, that core of it, where the protein synthesis happens, those bonds are getting made, is really conserved between organisms, and it's actually fairly slow compared to a lot of modern enzymes. Protein synthesis is, one of the slower processes that takes place in cells, and that's kind of an indication that this is something that got locked in very early. And I think that, if you think about that, the reason that something like that usually happens, if something, works well enough, but maybe not as fast as it could, the reason that something like that gets locked in is because the cost of changing it would be really too high for it to happen. Because if you think about what happens when evolution happens, is that cells or life is kind of exploring different solutions to the same problem, and so that happens usually through mutations. And so there's kind of experimentation that happens where you try something a little bit different. Most of the time it doesn't work better, some of the time it does, and that gives some advantage, and that's sort of how it proceeds. That's great, and it works for a lot of different enzymes and pieces of cellular machinery, partly because the loss of function in the course of evolving something might hurt a cell, but it might not be too deleterious, so that can kind of get explored. Protein synthesis is so core to a cell that breaking protein synthesis is fatal for pretty much any living thing. And so this is why the ribosome is kind of thought to be locked in time. The same, I think, is true, and probably even more so, of sort of the core of biochemistry. When you think about just the basic chemical structures that make up what goes in there, there's not really a chance to evolve that at this point, just because, again, what's the cost of changing it? There might be different amino acids that would work better, but life is pretty limited in the ability to explore that, because, again, if evolution is happening, usually that involves breaking things, with the chance that sometimes something works a little bit better and then it kind of continues to improve. But breaking something so core to life, like what makes up the DNA or RNA backbone, is pretty hard to do, and tends to kill the organism in which that evolution is happening. And so I kind of think of that as being locked in at this point. And so I think that if you were to roll the dice on another body that's really similar, again, it's possible that you certainly could get a different outcome. But like you suggest, you could absolutely get the same one again.
Yeah, but I think even to reach that point first, basically we need this one common ancestor molecule, which is what this RNA hypothesis or RNA world is, right? And that we can also define as LUCA, of course, in terms of biomolecules, that this is the one which gives the genetics later. The question there is that, so how strong evidence is for RNA as the LUCA, which is last universal common ancestor?
I think that there is evidence for RNA, certainly being really old. If you look at the core of the ribosome, so that protein synthesis machinery, that core part where the protein synthesis is happening, there's really not protein anywhere close to where the bonds are being formed. And so it looks like coded protein synthesis is, the function of a ribozyme. And so we know that or at least we have evidence that was a really ancient event in the evolution of life. In terms of what was explored beyond that, I think is a really interesting question. And I think that there could have been some tweaks certainly with that. But, going back to the origin of coded protein synthesis and ribosomal function, that was certainly a really early event.
Yeah, and here I think the other important thing is this structure-function relationship. So, of course, we get one string of RNA, which is this one letter code, but then once it gets transformed into a certain structure, that changes everything, right? So how relevant that would be? And are we also looking for that? Because this is also one of the reasons why there are so many RNA molecules present, at least in the current times.
Sorry, could you just clarify a little bit more exactly what you're asking about the structure function question?
So the fact that when we are talking about RNA as the major ancestor, right? Because RNA can fold into different structures, right? So are we also looking for certain folds which would be really important for for RNA as because there can be again two things: RNA as the storage device in a way that it is storing the genetic information. The other thing is the RNA as catalyzing the reactions. So are we also looking for folds which would be somehow separate these two things?
I think it's a really interesting question, and what you touched on with this idea of the RNA as a coding molecule and RNA as a catalytic molecule are two functions that I think it's convenient in terms of the emergence of life that they're present in the same molecule, but that sort of convenience of the dual function presents really its own set of challenges, and that for there to be a functional RNA that's going to act as a catalyst, it's going to be folded into some kind of three-dimensional structure like a protein, so kind of a globular structure. Whereas if you're thinking of RNA as an information carrier, it's going to need to be single-stranded, or at least accessible in a single-stranded form. And the reason for that is because to act as a template, that has to get read out by non-enzymatic polymerization or an early polymerase. And how that got solved early on, it is very much an active field of study, because if you look at how DNA gets copied, thinking about the principal information carrier in modern life, there are polymerases and in some cases additional enzymes called helicases, or the polymerases have helicase activity on their own. And what the helicases do is unwind the double helix to make it a substrate for these copying reactions so that they're accessible. The copying part of it, making an RNA that copies RNA, has been done, and it's something that continues to improve as people work to evolve these in the lab. The example that we have of an RNA polymerase comes from work David Bartel originally did on an RNA ligase. And since then, there's been a number of labs like Phil Holligers and Jerry Joyce and others that have evolved them to be able to copy better. But the one difference between these polymerases made of RNA and modern protein-based RNA or DNA polymerases is that they don't really act as helicases. They're more copying molecules. And so how you sort of solve this problem of the dual role or dual mandate for RNA as a primitive biomolecule is kind of a tricky one, because if you think about an early enzyme, you probably didn't have this sophisticated multi-domain thing. And so that's another thing where I think solutes again become kind of an interesting problem. If you look at some of the work that Jerry Joyce has done with some of the directed evolution of these polymerases that started kind of from this Bartel ligase, one of the things that they've done in some of their recent work, they were able to do what they called ribo-PCR, which is basically doing exponential amplification of RNA with one of these polymerases. And in the reactions that they had significant, hundreds of millimolar to molar quantities of solute added there. And that helps in some ways with processivity. And so I think that, having RNAs perform both of these functions without something like a helicase present is a tricky problem. And I think one where I suspect we're going to find a lot of the answers kind of at these interfacial questions where it's different polymers and solutes interacting with each other.
And to make things worse, I think we can also talk about solubility, because that's something interesting in a way, because I think when it gets folded, the molecules, they behave differently, right? And the fact that so to preserve that fold, it'll also depend a lot on the solvent in general. I mean, if you change the solvent, it may change the fold, probably change the catalysis. So how are we dealing with that issue?
Yeah, I think that it's a really good question. Thinking about this idea of an RNA in different contexts and whether it's going to be soluble is a really important one. And now when we're thinking about RNA and the ion environment or the salts that it's interacting with, inside a cell, the principal physiologic cation is potassium. And then the other critical ion for RNA to fold is really magnesium. And one thing that's gotten a lot of attention in the last 10 years or so has been work, especially from Lauren Williams's lab, on other ions that could have been present for RNA to fold and perform catalysis. And one that was certainly present in a soluble form that no longer is to nearly the same extent is iron. And iron is a really interesting example, because if you look at reduced iron, or that would be iron with sort of 2 electrons removed, what Lauren and some other groups have shown is that in a lot of ways this can substitute for magnesium, and ribozymes that need magnesium can take iron and use it to perform function. But the sort of downside of having iron around is that iron is especially good at promoting RNA degradation. And this is a problem that we deal with in modern life too. And this is one reason that, RNA has kind of, or it's thought that RNA has sort of been supplanted by DNA as the main information storage molecule. And that's that RNA is always this kind of ephemeral thing, and that it's always undergoing degradation and it needs salt to work, but salt is also promoting it falling apart. Magnesium will do it. And it turns out iron does that even more prodigiously, really, than magnesium. And so there's that degradation aspect of it. And then touching on the solubility thing that you mentioned as well, when you have these divalent ions in the presence of things like phosphate, they tend to precipitate as well. And so this is definitely a problem. I think having all the right things in the right place at the right time in order to get all this up and running.
But I think this this can also be solved by compartmentalization. Of course, then it is another problem, how do we get compartmentalization? But once we get it, I think then it doesn't matter what the outer solvent is. Somehow you can preserve the inner environment, right?
I think that I definitely agree that compartmentalization is really powerful and a phenomenon that life has embraced for a lot of reasons. I think that yeah, having a different inner and outer environment is certainly important. And then kind of I think something that flows from that is uh, something that's kind of basic to life is this idea of self and non-self, where you have the metabolism and biochemistry that's taking place inside versus outside, and that's sort of a necessary condition for having things like heritability that proceed, and certainly compartmentalization is one of the most straightforward ways to do that. Again, kind of thinking of it in terms of challenges and sort of what keeps us working is the fact that these are difficult things to get working together. You know, if you look at thinking about compartmentalization as modern compartmentalization, so where you have lipid bilayers like a cell membrane and components of sort of model prebiotic reactions in them. So for this example, thinking about RNA, we just talked about magnesium being important for RNA function and sort of having this downside of promoting degradation. The membranes kind of introduce another problem there too, and that's that if you have things like magnesium around that you need for the RNA to act as a catalyst, those tend to destabilize membranes. If you look at the membranes that are used as examples of model primitive membranes, those actually tend to be the most sensitive to magnesium. And so typically what people look at is, in contrast to a modern cell membrane, which is usually two fatty acid tails attached to a glycerol backbone, usually with a phosphate or some other head group on there. Those have some sensitivity to magnesium, but it's even more pronounced when you look at a model primitive membrane, where instead of two fatty acid tails, you just have fatty acids around, and these will form membranes. But the sort of downside is that in the presence of magnesium and fatty acids is, if you ever clean like soap scum off your shower, that's what soap scum is. So it tends to disrupt membranes. So this is another question that sort of presents two problems that are kind of at odds with each other. And again, I kind of keep coming back to the example of these interfacial problems and why they think that they're so interesting. I think that these are certainly the things that have to kind of get solved to get a better understanding of how life might have gotten started.
Yeah, and still when I think about it, I can't understand the link between, for example, genetics and compartmentalization. So how, for example, because one thing is, of course, there is this the lipids, they are forming the membranes, but then once we entrap certain genetics, which is some RNA molecule, for example, and then from there to get to the lipid metabolism, and then the fact that we need to maintain these compartments. So is there any link? I mean, how far are we to understand that part?
I think that the I guess what I thought of when you first mentioned coupling genetics to compartmentalization was what we touched on at the beginning about just self versus non-self, and that's certainly a key component. But then you mentioned lipid biosynthesis and other aspects of it. It turns out that getting fatty acid vesicles, so these sort of prebiotic like vesicles, to do things like growth and division turns out to be quite a bit more straightforward than for something like a modern cell membrane. So if you use something like a diacyl phospholipids, so basically the components that make up contemporary cell membranes, in contrast to something like a fatty acid membrane, so one made of something like oleic or decanoic acid, the diacyl phospholipid membrane, or the modern like cell membrane, is a lot less dynamic than the fatty acid membrane. And so in some respects that's good, and that's again thinking about why life has chosen or selected certain solutions to these problems. You want a stable membrane, and you want one that doesn't leak very much, and you want one that is only going to undergo division when you have specialized protein machinery that does that. And not having that presents its own set of challenges because there are reasons that life chose all this. But looking at these fatty acid membranes, because they're so much more dynamic, there are a lot of processes that can cause them to grow and divide. If there is osmotic pressure within fatty acid vesicles, so if there's more solute inside than outside, it'll tend to pick up water from the outside and grow. And so that's one mechanism by which growth can occur. You can have, because they're sort of semi-permeable, and not because of pores or anything like that, this is just intrinsic to having the sort of prebiotic-like fatty acid membrane, they can accumulate certain solutes from outside, but only certain things will go across the membrane. So if they undergo some kind of chemical transformation, they can go in but not out in some cases. And then in terms of the division problem, in contrast to a modern cell, when you have something that undergoes mitosis in this really tightly controlled process, if you look at a fatty acid membrane, under certain conditions you can have these membranes sort of transform from bubble-like structures into these thread-like structures. And then when they're mechanically disrupted, and that can just be blowing air across them, you can see them shear and retain their contents there. And so it's, again, I think, an example of where some of what presents problems in terms of getting life up and running with these really simple chemical systems also affords, I think, some opportunities to get these kind of lifelike behaviors in a sort of conceptually simpler way than we find in modern life.
Yeah. Then maybe now we can also talk a little bit about DNA. So going from RNA to DNA, basically getting this Watson-Crick base pairing, right? How important that is for, as in like storage device for the genetics?
I think that it's really important in terms of getting a modern type cell going, because if you think about how long a cell lives and sort of what it's doing during that time, you need a stable polymer, and DNA is a much more chemically stable polymer than RNA, because of that chemical change that's been done to it. And then beyond that, there's a number of things that make it advantageous to a living thing. If you look at DNA and how it's stored in some of the later products of evolution, a eukaryotic cell, it's stored in a nucleus, and it's tightly compacted around histone proteins that sort of let it get wrapped up, and you can store big genomes in a small place, basically. And then beyond that, just thinking about in terms of proteins recognizing and distinguishing DNA from RNA, that 2' hydroxyl on RNA versus DNA is a big chemical difference that proteins and things like that can recognize. And so in terms of chemical stability and function, you can definitely see reasons that life has selected it as the informational storage polymer. But I think thinking about sort of bootstrapping life and thinking about very small genomes and sort of what the minimal set of conditions are to get something going, it's certainly possible for, I think, RNA to play that role kind of in a limited way at the outset. But in terms of complexification of life and all the different functions that we see living things perform, it's not really surprising to me that DNA supplanted that information storage role of RNA for those reasons.
And actually, when we think about these wet and dry conditions in the prebiotic sort of conditions, the question there is that we can also think of other conditions like oxidation reduction, which would easily happen. So which one is like more prone to these changes when it comes to DNA or RNA?
They definitely. So I guess first with the wet dry cycles, I think that they, if you think about the reactions that assemble all the biopolymers that we have, if you think about DNA and RNA and protein, these are all formally dehydration condensation reactions. And so you can see, at least in terms of getting things started, how water removal was kind of important to that process or critical to that process. um, RNA, for some of the same reasons that it's a kind of fragile polymer, is also just a more dynamic polymer. And I can see how these might have been sort of easier to assemble. In terms of the redox chemistry, I think it's a really interesting question because, when we think about redox chemistry today, that's a function that is, when it's done by a cell sort of towards its metabolic goals, that's something that is usually performed by proteins and usually with cofactors or metal ions within an enzyme that are involved in shuttling it around. But then a second question of redox chemistry relates to the questions of damage. Cells are constantly undergoing different toxic insults that can range from chemical components that can poison enzymes or perform mutations, ultraviolet light, which can degrade things, or beyond that, or which can be created by that, reactive oxygen species. And both RNA and DNA are susceptible to this kind of type of oxidative damage, and they can certainly react with them. And we can see examples in life by which this type of process has been solved, or this type of problem has been solved. There are repair mechanisms for solving some of the oxidative damages that occur to genetic polymers in living cells. And again, I think this kind of becomes a timescale problem too, because if you think about, the timescale on which a modern cell versus what we imagine to be a potential primitive cell is expected to preserve its genome. The longer you have a genome around, the more you're going to have to solve these types of problems, because whether it's environmental conditions or metabolic processes within the cell that make these kind of oxidative things, you will need a polymer that's expected to last the lifetime of a modern cell to basically be around longer. And so DNA sort of solves that hydrolytic stability problem. And then I think, in modern life, we can see things that have kind of been tacked onto that to solve this problem of oxidative damage.
Yeah, but then I mean, since we know that this Watson, the structures can go beyond Watson-Crick pairing, right? And can that be a result of this oxidation reduction reactions?
Yeah, it's definitely an interesting question. I think that looking at some of the different structures that you can get outside of Watson-Crick base pairing, typically these are going to involve the bases making hydrogen bonding interactions with different parts of that polymer. And so, in a Watson-Crick base pairing, you have the Watson-Crick base paired with another Watson-Crick base. You can have cases where bases are making hydrogen bonds to the Hooks-Gene face or other face, the nucleic acid, or to the backbone or things like that. And this oxidative damage can definitely influence that. And that's very much, I think, a question that's interesting from a prebiotic perspective, as well as in modern biochemistry. There's been some really nice work looking at basically the products of oxidative damage of DNA. Gary Schuster's lab and others, Jacqueline Barton, worked on this for quite a while in terms of modern life, in terms of how it solves this problem. But one thing that comes out of that type of oxidative damage is that it often will change the types of interactions that the bases themselves can make. When you oxidize a strand of DNA, for example, what usually happens is that the DNA actually acts kind of like a conductor, and the missing electron will basically hop along the DNA, or the hole, they call it, will hop along the DNA until you get to a guanine residue. And that guanine residue will undergo a chemical transformation to a modified base called adeoxoguanine, and that will change the hydrogen bonding interactions that that G can make. And it actually won't influence the Watson-Crick phase, but it'll influence some of the other non-canonical secondary structures or alternative structures that it can make. And it looks like that could certainly play a role into what structures DNA or RNA can form. And so that definitely gets used in modern life. How operative that is in early life, I think, is a really interesting question.
Yeah, and using that, you guys have been building some tools around G4s for imaging some bio or life cells, etc.
Yeah, we're really interested in, I guess, G quadruplexes. So I guess for your listeners, these are 4-stranded versus the 2-stranded Watson-Crick structures that G-rich structures can make in DNA and RNA. And we're also interested in, yeah, as you mentioned, some of these imaging tools. So the idea with these imaging tools is that we would like to have fluorescent tools for following by microscopy or other fluorescent techniques, RNA in kind of the same way that we do with proteins. And it's kind of amazing that we have the tools that we do from proteins because, if you look back to the earliest, kind of most foundational fluorescent protein that we have that lets us do these types of experiments, it's called GFP, or green fluorescent protein. And it's almost kind of dumb luck that we have it. It's this protein that came out of a jellyfish. And I think the reason it was so lucky is that you can take that protein and express it in lots of different organisms. This jellyfish protein, we can take it and it works in a bacterium, it works in a fly, it works in a worm, it works all the way up to, mice and non-human primates. And the reason I think that's so amazing is that a lot of these proteins, when you try and express them heterologously or basically in a different organism than they were kind of born in, that often doesn't work. And why this is so remarkable in the case of GFP, I think, too, is that it's actually doing a post-translational modification on itself after it gets produced. So in the cell, GFP gets produced, like any other protein. It gets transcribed from DNA as RNA. The RNA gets read out by the ribosome, and that protein comes off the ribosome. But at that stage, when that protein comes off the ribosome, it's not actually green or fluorescent. It's just a protein. And what happens is there is this post-translational modification or a chemical reaction that it does with itself. And what happens in GFP is that 3 amino acids in the core of that GFP will react with one another. And that's what gives you that green fluorescent fragment. And so there's not a known natural RNA that can give you visible fluorescence like GFP does. And so that's one thing that my lab is working on as well, is trying to get tags that work for RNA imaging like they do for GFP. And so there's a number of other labs working on this that have been selecting different RNAs for doing this that can act as kind of these types of imaging tags. And it's a field that really has taken off in the last 10 years or so. We've known a few examples of these for a little bit longer, but it's really been in the last decade or so that there's been kind of a increase or surge of interest in exactly how these work. And so I think that I and probably other people that work in the field would concede that these are kind of the point where you're able to do some of these imaging experiments, but they're not to the point where it's quite as robust as we have with a lot of these fluorescent proteins, where we can take you or your listeners might have heard or seen the examples where you look at a palette of fluorescent proteins and you get kind of this rainbow color of spectrums. And we have those groups that have selected these have developed aptamer spanning kind of the full visible spectrum that can act as these RNA like fluorescent tags. it's not quite to the point where they're plug and play in a given experiment. You can just make a fusion with an RNA like you would with a protein, and it's kind of ready to go. We are able to start doing some of those experiments, but it's very much an active space where these continue to be improved, and we're working on developing some of these new tags that sort of enable those improvements and hopefully get us to the point where it is to the point where eventually you're going to be able to do exactly the same experiment you would with a protein and making a GFP fusion, taking an RNA and fusing it to one of these aptamer tags to do the imaging experiment.
Yeah, and then can these tags be used to study somehow the origins of life issues, like somehow to incorporate these RNA molecules into these tags into the other RNA molecules and see if like to look at it?
I think it's a really interesting question, and probably one that highlights some of a lot of the work that's been done on it in this kind of interceding 10 years between then and one of the earlier reports of these and now, and that's that looking at what we're doing with these, ultimately trying to study biological problems. I would say that the more challenging experiments, I guess kind of starting bad news first, has been to do the type of experiment you do with a fluorescent protein in whole organism or even a eukaryotic organism. There are some really nice examples with pepper and riboglow are two examples of these fluorescent tags that have been used to do eukaryotic imaging, mango being another example. But they certainly are more challenging to get working. And in terms of imaging, RNAs that there's not a whole lot of around, it's been hard. So then kind of going back in biological complexity to things like bacteria, they work quite a bit better. TJ Ha had some really nice work showing that you could use these to follow messenger RNAs. Still challenging. Looking at the way that this is done, usually the way that these experiments are done, you have fusions of RNA with repeats of some of these tags to increase the brightness of it. But then I think the example that has been incredibly productive, and we and other groups have done a lot of work on, especially in the last few years, has been using them as tags in some of these cell-free systems, because these are basically to the point where I think they are as mature as something like GFP in following things in cell-free reactions. For watching the readout of something like enzymatic in vitro transcription, you can follow these types of reactions. And so kind of going back a step further to what you mentioned about prebiotic chemistry, I think that these are absolutely tools that we can use to follow these types of reactions. Trying to think how long ago it was. It was probably 6 or 7 years ago. Kate Alamal and I and Jack Shostak's lab published a paper looking at non-enzymatic RNA polymerization and ways to use that to generate functional RNAs. And one of the things that we were looking at with that was the earliest fluorescent RNA that I know of, and that was an RNA that binds to a dye called malachite green that was originally selected to make self-cleaving RNAs, and we found out after the fact that it actually acted as one of these fluorescent RNAs. These types of non-enzymatic reactions were basically copying just a few bases of RNA. So we were using copying chemistry that went fairly slowly and was kind of a model of some really early copying reactions, and slowly enough that we weren't able to get it to work with reading out something like the full length of that aptamer, which would be, I forget the length, but 50 to 100 bases. What we were doing in this was looking at basically closing a stem in a stem loop in that RNA, and looking at that closed stem by activation of the fluorescent RNA. And that actually worked really well. And so I think that kind of showcases the examples of things that you can study with this, because if you're looking at something where you have sort of a primitive RNA copying reaction where, we're just getting through a few bases at a time, something like this that can give you a fluorescent signal, I think is a really powerful tool for reactions like that.
Yeah, it will be amazing especially if we think in this exobiology setup. For example, if I don't know if so first probably we can talk about this thing that how we get information from Mars. Let's say that once we send our robots and etc. So what sort of information we get, and have we ever managed to get a sort of sample, probably soil sample, to Earth, or no?
So that is, yeah, I think kind of the $10,000 question as far as Mars is what exactly is it going to look like when we do sample return? Because the experiments that have been done to date have all been done in situ. And so the way that these have worked up till now has been, you know, putting some kind of instrument or spacecraft on Mars and having some analytical instrumentation there, and using that to kind of analyze it in place. And we've learned a lot from that about the geochemistry of Mars, everything that we've been talking about the composition of regolith. But necessarily when you do an experiment like that, you're kind of limited in terms of the level of instrumentation that you take, because just thinking about logistically, what you're doing with that type of experiment is you've basically got to get it from Earth to Mars. And so you're launching it from Earth, and then it goes through Earth's gravity and through Mars's gravity, and it takes over a year to get there. And so because of those constraints in transportation, we're kind of limited in the questions that we can ask, because you've got to pack light for a trip like that. And so it would be really powerful to do a sample return experiment and get some of that back. And so last year there was several missions that launched for landing on Mars, some of which have landed now, that are collecting samples and caching them for eventual future sample returns. So hopefully if we have this conversation in a few years, we'll have learned a lot from that, because I think that that's the type of thing where you really can't beat that, and you can't replicate it, I think, with any of these types of in situ experiments, because coming back to the example of the Murchison meteorite that we were talking about, just looking at what was present when it first landed versus what's present or what we're able to do now, the type of analytical chemistry that is possible, and this is on Earth, just in terms of advancement of technology, we're able to analyze what's there with a lot more sophistication than we used to. The same was true of I think your listeners will probably be familiar with the Miller-Urey experiment. Stanley Miller had an experiment in his lab in Urey's lab, where he took some precursor like molecules and basically introduced light, or introduced energy, so light and spark and things like that. And in the early experiments, they found a subset of amino acids, and then when it was analyzed years later, with more modern analytical techniques, they were able to find a whole suite of amino acids and other biomolecules that kind of weren't there in the first place. And so I think what you touched on is absolutely right. The sample return is going to enable us to do a lot of things that just aren't present and possible, because it's simply with existing technology not possible to do those experiments with something that's basically gone through launching and landing there and has to land in a functional state, and all the different constraints that go into that type of experiment.
Yeah, but then how are we receiving the information now?
So the experiments right now are done on the surface of Mars and transmitted basically back to Earth. So the experiments are essentially being done by equipment that's landed there, and then that is radio transmitted basically back to Earth. So essentially all that we know right now that's been done has been done on the surface, and the only thing that's coming back is the information.
Yeah, and this is through a mass spectrometer, right?
Yeah, mass spec is probably I would say the core piece of instrumentation that they've used in doing these.
Yeah, but then maybe in the future missions, if we can also include some, I don't know, some spectroscopy to image the molecules and to see if that will be possible. I don't know. Maybe.
So yeah, there definitely is some spectrometry capacity in terms of the instrumentation that's there, but in terms of the type of thing that we would run on Earth, in terms of even in terms of the mass spec, a lot of the time you would do something like separate out the mixture with a liquid chromatography instrument, or the when we say spectrometry, that can refer to something as, I guess, diverse as, something like light spectroscopy, all the way out to something like NMR that gives us, direct kind of structural information. And I think you're absolutely right. If we could get that information, that would be really powerful.
Yeah. So what are your major questions now that you want to continue or focus on?
What we would like to look at is, or what we are looking at is, the knowing what we know about the surface of Mars from experiments that have been done, and looking at some of these brines. What are the consequences of having those around in terms of, if we're looking at a prebiotic chemistry situation, what's influencing that and what might let that look like? And I think that it's a really interesting question to me because it touches on, I think, questions that are relevant to the emergence of life on Earth, assuming, this all happened here from ingredients that we had here and there wasn't any kind of transfer or seeding of life. And what we have in extant life would then be kind of a fossil of the chemical conditions in which life emerged. Because if we're looking at features of RNA and DNA that have been preserved throughout the early evolution and modern evolution of life, that's had to be, the function there has to be able to survive and function in whatever solution conditions and other environmental conditions generally that were present. So salinity, geochemistry, temperature, and all that. And so I think that Mars, for the same reasons, I think it's a really interesting environment to look at for whether life exists or existed. I think is a really interesting environment to look at prebiotic chemistry, because again, it's similar in so many ways to Earth, but I think different in some important ways. Looking at the modern surface of Mars, and this has certainly changed over time as I think availability of water in the atmosphere has changed. But you've got regolith that in many ways looks a lot like that on Earth. There's at least towards the surface, probably transient liquid water. There's a lot of salt around in whatever condition that you have there. Then beyond that, there's kind of a whole different set of challenges as well. Ultraviolet radiation is really difficult. The UV fluxes on Mars, or cosmic fluxes, are really high as well. And so thinking about exactly how that might look in terms of what's allowable for evolution of life, I think are really interesting questions. And I think that that's a question that is interesting for essentially any celestial body. If you look within our solar system or even at exoplanets, we have a collaboration looking at exoplanets near M dwarf planets. The same kind of things are going to arise there. Whatever kind of biopolymers emerge, we're working within the constraints of chemistry. It's the same set of elements, and so things like ultraviolet radiation, cosmic radiation, all kind of tie into this, and how those influence what's allowable to emerge and what could last through time are questions we're very much interested in.
Yeah. And what about populating Mars with microbes?
It's a really interesting question, and I think that it's one of these where it is, I think that different groups have goals that are at odds with each other in terms of this, because if you're thinking about, do we want to solve or if we want to solve the problem of, is there life on Mars, the absolute last thing you would want to do is put more stuff up there, because that's presumably going to ruin whatever is there. That's you had mentioned, if life from another planet lands on Earth, it's so hard to do the experiments to determine whether it's life from another planet or life that's on Earth, because there's tons of life on Earth, and the same kind of chemistry is going to be happening there. And I think that for me at least, the question of whether life exists or is evolving on another planet, probably right now a more interesting one to me than could we do something like put microbes and terraform Mars. And I think that part of it is, my own biases in terms of the experiments that I'm interested in doing and the things that I think about. But beyond that, if we think about microbes, and I realize you didn't say terraforming, but a lot of the time that's what's involved, is thinking about them in terms of making a habitable Mars. However well that works, it's certainly going to be a lot harder than preserving what we've got, because if we think about environmental issues on Earth, and things that we're thinking about with climate change and EV fluxes and carbon cycle here, and we're tremendously concerned about small tweaks to a cycle making something that was habitable in the way that we knew it 50 years ago looking significantly different than it does now. And this is kind of a small change at the edges here. I think that the type of energy that can get devoted to solving a problem like that is probably better dedicated to something on Earth. And that's not to say that it's not an interesting problem, but I think that it's probably an easy, more immediate one that's not easy by far, I guess, on Earth right now.
Yeah, actually, Jeff Hawkins talks about these issues in his book, A Thousand Brains. That they, So this is what he says, that should we colonize Mars? I mean, of course, that's a question, and of course, there are many aspects to it. And at the end, it may happen that we let's say manage to colonize Mars, and if something is coming to Earth, and then a lot of Earth people, they want to go to Mars. And of course, then there can be conflicts, and we end up killing each other.
Right.
So these things are always there, but the point is that, again, I mean, if we are intelligent enough, we should be able to figure it out, right?
I think it would be really cool, if we're able to do it. When I come back to this question, I can't not think of the example on Earth and how hard a problem it is right now. And I think if you ask me in isolation, would I like to see that? Of course, I would love to, but I immediately go to the similar problem that we have right now whenever it comes up.
Yeah, exactly. Yeah. So what else do you recommend to read for the people who want to read about the origins of life?
I think that I'd say the last 5 years or so, I have found so many good papers and discussion of a lot of this on Twitter, and it's taken off in a really profound way during the pandemic. I think just in terms of investigators, Donald Burke does some really exciting work. Sarah Walker at ASU, Batul Kazar, Mark Ditzler at NASA Ames, and I mentioned Twitter and some of the discussion there in their papers as well, but all of those authors that I mentioned too do a lot of public outreach as well. One thing that I touched on too at the beginning was talking about this PCE3, this RCN program that NASA has. PCE3 is one of those that I'm involved in, but RCN, the Research Coordination Networks, if you search for that in the context of NASA, there's a few of those that are interested in some of these questions of early life, and each of those has kind of a public outreach component too. Another thing I like too is TED Talks. Penny Boston gave a really nice TED Talk on life on Mars, and there's been a number of other investigators that have given some really nice talks in that space too. So I don't think I mentioned a single book in there, but these are all kind of online resources that I think are great places to look.
Yeah, that's great. I mean, so if we think about origins of life research, few decades back, it was all like cocktail of stuff, and now it's getting, I think, more directed in a way, especially with this molecular evolution and all these new methods that now people are like looking at origin of life research completely differently, right?
I think it's been a really exciting time to work in this space. I totally agree, and that there's always been cross-pollination, I think, between people that are doing molecular biology and people that are doing physics and geology and astronomy. That where there is overlap between the fields. But I completely agree. I feel like, in the last 10, 15 years or so, there really has been an explosion of really interesting collaborative projects that have come out of this. So I'm really excited to see where it goes.
Okay, so thank you so much for accepting the invitation, coming to the podcast and discussing, sharing your ideas, your research. Thank you.
Thanks so much for having me. It was a lot of fun talking with you today.